I misunderstood the question, so I wrote the entirely wrong answer.
It happens. It happens to the best of us.
It really does happen.
It does. It does. I'm not lying. I don't lie to y'all.
Okay, this, and then I have the wrong coefficients here. For the climb,
Yeah.
everything else was right. I knew everything else was right. It was just zero. The 5 formulas of this basically standard, like add like 5 percentage points.
Yeah.
Should I just leave it at like the page, or should I just
Leave it at the page, and then I'm gonna remember. I'm gonna remember. I'm gonna put it in here. I'm not gonna put it with the others. That way I remember. Okay. I need. Sure.
That's really bad on this one.
Oh my God, I did this twice.
I also on the
Sorry.
No, it's okay. It's really not that serious. But no, but it's really fun.
You got it right.
This says. Oh,
Okay,
for
that's
4
right.
points.
So it's
Name for 2. So it's fine. And that's fine. I don't know. Is that right or is that wrong?
No, that's the right answer.
Okay.
That's just.
It's really fine.
That's just.
I'm getting it. It's fine.
It's okay. It's okay. It's okay. But you need your point. Okay. I don't want the interactive notebook. Did I click the wrong thing? I probably did. Animation. Thank you. All right. Why does it keep going to the ebook? I am clicking on the animations, right? No, I'm clicking on the ebook. Like 3 times I've clicked on the ebook. Okay, and then I need right clicker.
Yeah, I'm not going I'm also
molecular animation.
I will not be studying here because I'm just gonna take an hour.
Okay. We need to go all the way. We were here, right? We were bioenergetics.
We were right about to do math, and then you quit.
I think we're gonna quit. I think we're gonna skip the math just so that we can get overarching ideas.
There will be no math.
No, we're skipping this slide. Like this,
Okay,
the
but
math from
is there
this
math
slide.
Part of this on the front.
This formula, no.
Oh, okay.
This formula, no.
I don't know what delta beside it.
I feel like
That's the potential. That's the electric potential. Well, obviously, because you only have the pH and the electric potential. I know what delta pH is, so.
That's exactly right. Okay, I feel like I haven't logged into something, but maybe I did.
Whatever.
It's clicker. Okay, I opened it, but I didn't log in. Thank you. See, I was like, I knew there was something.
How do you guys do that? I don't understand. Get more sleep, please.
Yeah, you really do need to get more sleep.
yeah,
Okay,
I've been sleeping 8 hours every night.
so attendance should be rolling.
I got 8 hours until it's like 13.
Go ahead and check in on attendance. Please go ahead and check in on attendance.
It's fine. We're not here about that.
Do we get like points for coming to class every day?
No, but you don't get kicked out of the class because you don't show up. So that's a good thing, right? That's a good thing. If you miss a certain percentage of the class, then it can be an automatic F. So yeah, like, you know, you did show up to class. Yeah, you know, it's cool. It's cool. So check in on attendance. All right, y'all. I love today's joke. What do you call a metric cookie? A graham cracker. Yeah, the final in this class is tomorrow.
Yeah.
Right?
Well, like I've been studying, but like I don't know what day it is.
Yeah, I feel you on that one. I don't know what day it is. I feel you on that one. All right, so we do have a final exam tomorrow at 1 o'clock, right? 1 o'clock, 1 to 3, right here. So what we're gonna do is we're gonna skip the math here, which means I'm gonna skip the math on the exam because I really want you to have a complete and whole picture of kind of what happens. I want to get to ATP production, right? Why do we do glycolysis? Why do we do citrate cycle? The whole point is to do what? Make ATP. That's the name of the game. So I really want us to make ATP today. All right. So before we do that, let's like talk for just a minute about what we've done so far in the electron transport chain. Structurally, when you think about the electron transport chain, what must the electron transport chain have? It must be able to function not only in the inner membrane, right, but also in the matrix. So when you think about these protein complexes that are embedded in the inner mitochondrial membrane, why do they have to have a functional component in the inner membrane and a functional component in the matrix? What's up with that? Why do you need the matrix part of it? What's in the matrix? That's where the citric acid cycle happens. So that's where you made FADH. That's where you made FADH2 and NADH, right? So we have to have a functional component that faces the matrix. Absolutely. Now, why do we have to have a functional component that works in the inner membrane space?
Because that's 5% of the electrical space.
No, it could be a free floating complex that does that. Doesn't have to be embedded in the membrane. Why does the electron transport chain have to have functional components embedded in the inner mitochondrial membrane? To where? That's right. So you can transport those electrons to your carrier molecules. Your carrier molecules are molecules that can move around within, guess what? The inner membrane space, the inner membrane, not the inner membrane space, but the inner membrane, right? So you have to get those electrons from complex 1 to complex 2. That's actually wrong. From complex 1 to complex 3 or complex 2 to complex 3, right? Whatever the connection is, you need to be able to move those electrons from one complex to another complex. Well, if your mobile electron carrier is embedded in the membrane, guess what? You need a functional component that's embedded in the membrane. So I just kind of want y'all thinking about that and understanding those things. So before we get into ATP synthase, what I want to do is I want to ask you, what have we made so far? If I have an NADH molecule, what have I done with that NADH molecule up until today with the knowledge you have? I took 10 hydrogens and I did what to them? I pumped them from the matrix to inner membrane space. Do you remember you'll see this notation in the book a lot. Do you remember that we said the negative part and the positive part? Do you remember that? So a lot of times you'll see it written this way. Okay, so how about FADH2? Do we still get 10? No, because we skip complex 1 and we go to complex 2, and complex 2 doesn't pump, and complex 1 pumps 4 hydrogen ions. So 10 minus 4 is 6. Right? That's going to be really, really important later. So like put that on the side. We're going to come back to that. Okay. So, oh, but I want to go back here. Okay. So what do we do with those hydrogen ions? We've got them on the inner membrane space of the mitochondria, right? What do we do with them? Well, we want to send them through the ATP synthase complex, right? Now that ATP synthase complex we used to call complex 5. but it's not a complex. Why is it not a complex? It's the electron transport chain. And our final electron acceptor was oxygen. Are electrons moving anymore? No, we're done with electrons. We don't need those stinking electrons anymore right now. So we've shipped them to oxygen. Oxygen's turned into water. We're done with electrons. Now what are we dealing with? Protons. We're dealing with protons, with hydrogen ions, right? So it is not part of the electron transport chain because it's not doing anything with electrons, right? But we didn't understand that in the beginning, you know, like most things. So this is the ATP synthase complex. It is highly, highly conserved between prokaryotes and eukaryotes. So there are some things missing in bacterial versus eukaryotic, and this is the simple, one of the simplest eukaryotes is yeast, but very, very highly conserved. So there are two ways to divide up the ATP synthase complex. One of them is a structural division. The other is going to be, can you guess? Functional. Okay, so let's talk about structural first. We have two basic structural components. We have the F1 component, and this is basically F1, everything that is poking out into where? The matrix. So F1 is in matrix. Then you have F0. F0 is embedded in the mitochondrial inner membrane. Okay. Now, F1 is where the action happens. This is where we make ATP. But what do we need in order to make ATP? We have to take our hydrogen ions and allow them to flow down their concentration gradient. Well, in order to do that, I need a channel, right? So my F0 acts as the proton channel that crosses that intermembrane space, that intermembrane, not the intermembrane space. Why am I doing that today? Don't know. So our hydrogen ions come through here, and as they're going down their electrochemical gradient, they can drive a rotor within the ATP synthase complex, which changes this catalytic subunit, which actually produces ATP. Okay, so this is the structural categorization, right? But now we're going to talk about functional, right? So these are our functional subunits. So you have 1, 2, 3 functional subunits. Okay, I want you to kind of think about this kind of like a motor, right? So like if you have to, you've seen like the pedal operated things like a boat or a bicycle, anything that you have to power, right? You have to make something turn. The thing that we're trying to make turn is called the headpiece. The headpiece is where we make ATP. This is the action, right? This is the rotor. So when this rotor moves, this part that sticks down actually sticks down right in between all of the pieces of the headpiece. And as this rotor moves, it bumps into each of the individual subunits inside of the headpiece, and it induces a conformational change, right? That's what drives everything in biochemistry, is that and phosphorylation, but phosphorylation is going to change make a conformational change anyway, but it's a conformational change, right? And we know when things change shape, they change function. So that's how we drive ATP synthesis. And then we also have what's called the stator. So this is basically holding everything attached, right? So before I talk about all the details of this, I want to show you the animation because they have an awesome animation for this. So I'm going to switch over. I'm going to show you this. I'm going to try not to blast you out, but I make no promises.
Consists of two large structural components.
Is that good?
F1,
Yeah, sounds good. Okay. I'll start at the beginning.
the mitochondrial ATP synthase complex consists of two large structural components. F1, which encodes catalytic activity, and F0, which functions as a proton channel crossing the inner mitochondrial membrane. In yeast, the mitochondrial F1 component consists of 3 alpha subunits, 3 beta subunits, and 1 each of gamma, delta, epsilon, and OSCP subunits. The F0 component in yeast contains 1 A subunit, 2 B subunits, and 10 C subunits. It's useful to think of the ATP synthase complex as 3 functional units. The rotor, made up of gamma, delta, and epsilon subunits and the C subunit ring, rotates as a single unit. The catalytic headpiece, containing a hexameric arrangement of 3 alpha and 3 beta subunits, synthesizes ATP. And the stator, which consists of the A subunit embedded in the membrane and an immobile stabilizing arm made of the B, G, H, and OSCP subunits. The F1 component's beta subunits each contain a catalytic site that can catalyze the formation of ATP from ADP and inorganic phosphate. At any given time, each catalytic site is in a different conformation and has a different affinity for ATP. This results in one site generally being empty, called the open conformation, one occupied by ATP and inorganic phosphate, called the loose conformation, and the last one, referred to as the tight conformation, being occupied by ATP. The rotation of the gamma subunit causes the change in conformation in the beta subunits. As the gamma subunit rotates in a clockwise direction, the beta subunits undergo sequential conformational changes, each going from loose to tight to open conformations. Note that when viewed from the matrix, the gamma subunit of the intact complex appears to rotate in a counterclockwise direction. In the yeast mitochondrial ATP synthase, each 120-degree turn of the gamma subunit is powered by the movement of approximately 3 protons through the F0 subunit. How does proton movement cause rotation of the gamma subunit? The 2-channel model proposes that the A subunit contains 2 half channels that provide access to the C subunit ring from either the intermembrane space or the mitochondrial matrix. According to this model, a proton from the intermembrane space, where proton concentrations are highest, first enters the half channel. The proton then comes in contact with a negatively charged aspartate 59 residue in the C subunit. The proton neutralizes the negative charge, allowing the C subunit ring to rotate in a clockwise direction when viewed from the intermembrane space. On the other side of the C ring, a proton bound to an aspartate 59 of a different C subunit gains access to the second half channel and enters the mitochondrial matrix, where proton concentrations are low. Using this extraordinary protein machine, cells are able to harness the energy of a proton gradient to drive the synthesis of ATP.
Isn't that like awesome? It's just so cool and amazing. So you do have to kind of know some of the subunits, right? So alpha and beta, that's what's in the headpiece. This is what's actually changing conformations and driving that ATP to form, right? But what causes those conformational changes? The rotor, right? The rotor turns, and as that end of the rotor hits each of the individual subunits, they change conformation. Well, what makes the rotor turn? Let me do, oh, that's not what I wanted to do. Full screen. Okay, so the C subunits, right? The C subunits, when they come through, when the hydrogen ions come through the half channel, right? They can bind to a a negatively charged residue in the C units, which neutralizes it, which allows it to freely rotate, right? Then it drops it off and it goes through the second half of the half channel, right? And that's how we go down a concentration gradient, but drive ATP synthesis. Okay, we did that. So protons are flowing through the component that's embedded in the membrane. This is the structural F0 component, right? It's the F1 component that has the alpha and the beta subunits that undergo the conformational change from loose to tight to open, right? So when that happens, every time the rotor pulls 3 hydrogen ions, right? So this is, the rotor rotates 120 degrees per 3 protons, right? So when that happens, you got to think about, if I want to go 360, how many times does the rotor have to turn? 3 times, right? 120 times 3 is 360. So that means how many different conformations could I possibly have? 3, right? So my first conformation is when I have ADP and inorganic phosphate bound. So what did they call this one? The loose, L-O-O-S-E, loose, right? Okay, so then we will catalyze the addition of the phosphate to ADP, and we will make ATP. This is the tight. Then ATP has to be released. So that is the open. Then ADP can bind with PI, that's the loose. And you see how we're rotating through again? Makes perfect sense, right? All the math works out. Isn't that so cool? All right, so this is, if you are looking at the alpha and beta subunits from the matrix side. So if we're from the matrix side, we're looking up at the ATP synthase complex, right? So you're looking at the bottom. In the video, it started with it looking down. So you got to think clockwise and counterclockwise, right? So if you're in the open conformation, you've released ATP and you're getting ready to bind ADP and inorganic phosphate, right? So let's just track what happens to this one subunit, this one beta subunit. So when 3 hydrogen ions flow through, now we're going to go from being empty to being bound to ADP and inorganic phosphate. Now that rotor is going to rotate another 120 degrees. And what do I have at that same subunit? ATP. So if you just follow the same subunit for 3 rounds, you see the 3 different conformations, right? And it's all about how this rotor is interacting with that subunit, what conformation it's in, what it can actually do. Kind of cool, right? Kind of insanely cool. Okay, so this was a really interesting experiment, and not just because they did it with actin, which makes it a little more cool, but what they did was they took the catalytic part of the ATP synthase complex and they attached it to a solid surface, like a glass slide, right? And they attached an actin filament to the rotor, right? The gamma subunit. This is the thing that induces the conformational change in the alpha and the beta subunits. And they added a magnet to the other end of the actin filament. So now if I have a magnet underneath the slide and I have a magnet on the actin, as I move the magnet under the slide, I can pull the actin around whichever way I want, right? Okay, now here's the cool part. So when they did this, if you clockwise rotated it, right? If you turn that clockwise, you hydrolyze ATP. If you turn it counterclockwise, you make ATP. Isn't that cool? Okay, I love science. It's all right. I can be impressed with it, and y'all don't have to be. But it's an amazing system. So before we talk about transport systems in the mitochondria, I want to talk about something else first. So what have we done so far, right? What did we start with in metabolism? Like glycolysis. So what was our starting subunit in glycolysis? Glucose. So in your textbook, what does it say you can get from one glucose molecule? No, maximum at the end. We're going to start at the start and go all the way to the end. We're going to make ATP. How many? 32 ATP. Where did that 32 ATP come from? Right now, you can calculate that. You ready? Let's do it, right? We need some conversion factors, right? What kind of conversion factors do we need?
Yeah, what do you
What do I mean by that? I mean that I need to know how many ATP I can get per energy carrying molecule, right? So I'm going to tell you this. It's okay. I'm going to tell you this. You can go through and calculate it, but I'm going to tell you this. You can get 5 ATP per 2 NADH molecules. Can we get the same amount per 2 FADH? 2? No, we can actually get 3. Okay. If we go through the full, I'm going to put a little asterisk. If we go through the full ETC, right? I'm assuming that we go through the full ETC. All right. So what happens to glucose? What's the first thing we're going to go through? What first process? Glycolysis, just in general, but you're right. That is the first step, right? So we have one glucose molecule. Oh, you see it, glucose molecule. There are things that happen to that glucose molecule, and what do we get at the end of glycolysis? 2-pyruvate. 2-pyruvate. Okay. Come on. Oh, I'm not. I got to extend the page. Okay. So what happens to those 2-pyruvate? We want to convert them to acetyl-CoA. So this glucose was in where? The cytosol, right? So where are the pyruvate? In the cytosol. So where do we need to ship it? Mitochondria. So I'm going to come down here, and you're going to see why I do this in a minute. But I'm going to do this, right? So everything kind of above is in the cytosol, and everything below we're going to be in the matrix. This is a mitochondrial matrix. This is the cytosol. Okay. What did we get out of glucose when we went from 1 glucose to 2-pyruvate without, I'm not talking about the pyruvate, but like what was our net outcome from glycolysis? What did we get? We got 2 ATP. What else did we get? 2 NADH. But oh no, NADH is in the cytosol. That's not good, right? Where's the ETC? In the matrix. Do we have a problem? Yes, we have a problem. So the cool thing to know, right, is that we also create NADH in lots of other cellular processes that we're not even going to touch on this semester, but please take biochem too. I'm going to plug myself anytime possible. So these 2 NADHs, I've got to get them into the matrix. There are 2 different ways that I can get them into the matrix. They are both shuttle systems, which is like the next topic that we're going to talk about. We haven't talked about it yet. So if you're in the liver, you're going to use the malate, malate shuttle. And when you use the malate shuttle, you are going to get 5 ATP out of that 2 NADH, right? Because that was our thing, 5 ATP from 2 NADH. That's the liver. Now, in muscle cells, we need ATP now, and we need lots of it all the time, right? You're constantly moving, you need more ATP, more ATP. So we actually sacrifice a little bit of that energy in order to be able to make ATP more quickly. So we use a different shuttle system, and we only get 3 ATP out of that same 2 NADH molecules because it's a different shuttle. It's a glyceraldehyde 3-phosphate shuttle. It's less complex, and so it takes less time, but you don't harness as much energy from it. Okay, so we're going to make a different amount of ATP whether we're in the liver or we're in the muscle cells. Okay, all right. So then these 2 ATP really end up as 2 ATP, right? Okay. All right. So now extend the page. Keep extending the page, right? Okay. So now we have, what do we have? Pyruvate. What happens to pyruvate? We're going to convert it to acetyl-CoA. How many? 2 acetyl-CoA. What else do we make in that reaction? CO2, but that's kind of junk, right? It's kind of junk. NADH, how many? 2 NADH. Okay, so now these 2 NADHs, if they go through the full, right, full ETC, how many ATP am I going to make from them? 5. Okay, now my acetyl-CoA. Acetyl-CoA is going to go through what? Citrate. Citrate. The citrate cycle is going to make what? Gotta extend the page again. My goodness. Now be careful. Be careful. It's 2 acetyl-CoA. So 6 NADH, 2 FADH2, and 2 GTP, which is really like 2 ATP, right? So then how many ATPs do we get from the FADH2? 3. How many do we get from the NADH? 15. Okay, so now I want 2 answers. I want to know in the liver, what do I get? And in the muscle, what do I get? So tell me muscle first, because I'm in red right now. So you add up all the black and the red. In the muscle, not 32, 30. How many do we get in the liver? 32. Cool, right? From up here? 5 ATP from 2 NADH and 3 from 2 FADH2. Isn't that kind of cool? Right? So a lot of times you see this in the book. Okay, 1 glucose molecule gives you 32 ATP. How? Now you know how, right? Didn't you? You were just dying to know. I know that y'all were just dying to know.
Under what conditions did you say that it'll take the liver mechanism versus the muscle? Or?
It depends on what tissue type you're in.
Okay.
What enzymes are available for that exchange.
Okay.
And I'm going to show you why that exchange, how it works. I'll show you today. But it depends on tissue type, because it depends on the energy needs of the cell, right? In the liver, you're not going to need that much energy that quickly. But in the muscle, I'm willing to give up a little bit of energy to get it back really quick, right? You think this is a good what?
I think so.
Yeah. No. My gosh, no. Okay, wait, let me go back. Okay, right? So this is where these 2 transport systems come in, right? Once you make that NADH in the cytosol, you can't undergo glycolysis anymore until you regenerate the NAD back to NAD+, right? So how are you going to do that if the things that pick up the electrons are actually in the mitochondria? We need a shuttle system. We need to be able to transport those energy-carrying molecules into the mitochondrial matrix. They can participate in the electron transport chain, and at the same time, you regenerate what you need in order to go through glycolysis again. So we have 2 different systems. Malate shuttle system is for the liver. The glycerol 3-phosphate is for muscle. So instead of actually transporting the molecule through a pore, they're electron carrying molecules, right? So why can't we just pass the electrons on to other molecules that can then be transported, that are normally transported, right? So we're kind of just taking advantage of what's there. So you have to think about that kind of transport. Then you have to think about, okay, I'm making ATP in the mitochondrial matrix. So any substrate in that reaction is going to have to be pumped, guess what? Into the matrix. Okay, so I need to pump ADP and inorganic phosphate into the matrix. But I've just made ATP in the matrix. But where do most of the reactions that a cell uses energy for occur? in the cytoplasm. So if I made ATP in the matrix of the mitochondria, but I want to use it in the cytoplasm, what do I have to do? Transport it. Gosh, we've been transporting a lot of stuff now, right? It's okay. So let's talk about the ATP and ADP first, because that's kind of a little bit easier. So we talked about this before, that there's a translocase. So it exports one ATP molecule for every ADP that is imported. Then it kind of makes sense in terms of charge, and I'm going to tell you about that in a minute. And then you have the phosphate translocase. So it's a different pump that translocates your inorganic phosphate, and it can transport it against a proton, a hydrogen ion, or it can transport it with a hydroxyl group, with an OH. So I'm going to show you both of them. Okay, so this is the one that will translocate ATP and ADP. So they call this the banana model because it's the fluctuating banana model, because it looks like this, right? So we have the inner membrane space up here above me, we have the matrix down here, and they're sitting here like this, right? So an ADP will come in and bind. When the ADP comes in and binds, right, it's facing the cytosol. It's going to undergo a conformational change, which then allows it to open up to the matrix side. So we call this the M conformation because it's the matrix side. We call this the C conformation because this is the cytosolic side, right? So it's cytosolic, it binds an ADP. No, that's a good name. This is a good name. What? Kind of does look like a W, but then so does this. Looks like an M. I don't know, but anyway. So you you turn into the mitochondrial conformation, right? You drop off your ADP, but before you can face back to the inner membrane space, what do we need to pick up? An ATP. So we pick up an ATP that induces the conformational change that opens it back up to the cytosol. Right? So it goes CM, 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 back and forth, back and forth. So the other thing that's kind of important about this membrane transport protein is that you have to kind of think about charges. So if you have an ADP molecule, how many negative charges, right? Because that's what, when you phosphorylate something, when you add a phosphate group to it, that phosphate group is negatively charged, right? So ADP has how many negatively charged things? 3. It has 3. Because you have one phosphate that has 1 negative charge, you have one phosphate that has 2 negative charges. The one on the end has 2. So now ATP, for each phosphate, you have a negative charge, and then the one on the end has 2, so you have an overall charge of -4. Okay, now, what's the overall charge in the inner membrane space? It's positive, right? So we can actually kind of balance the charge of the inner membrane space by using this pump, right? Because I'm taking something that is a -3 and bringing it in, and I'm taking something that's a -4 and pumping it out. So I now just added a negative charge to the inner membrane space. So it works better because of that, right? So and you don't have to worry about these. These are inhibitors, and there were experiments that were shown that this is how it works, but that's fine. As long as you understand how the mechanism works, I'm good. All right, so the other thing that we need to make ATP is inorganic phosphate, right? So the inorganic phosphate can either transport through a symporter or an antiporter. What we've talked about so far was it being a symporter, right? Because if you're transporting one inorganic phosphate group, it's actually conjugated to 2 hydrogens, so it has a charge of -1, right? Because it normally has 3 negatives, but if you put it with 2 hydrogens, now you have a charge of -1. So if we transport them at the same time, what's the overall charge of the 2 things that we're simultaneously transporting? It's a net of 0, right? A -1 and a +1, net of 0, right? What you can also do is you can do a antiporter where we don't change the charge, again, of the intermembrane space or the mitochondrial matrix, because we're going to take our negatively charged inorganic phosphate, pump it in to the intermembrane space, and then we're going to take a hydroxyl, right? Why do we have these floating around? What's up with that? Water, where do you think we got all of our hydrogen ions from? Our protons? From water, right? So we have these floating around. So depending on the pH, depending on what the cell needs, it's going to use one or the other. So if we pump it this way, if we pump it opposite the way that we pump the inorganic phosphate, we balance out the charge and we have no charge difference. Okay. So this is just taking kind of all the pumps in together. So here's our Atp synthase complex, right? We need ADP, we need inorganic phosphate. Our ADP comes from the Atp translocase. Our inorganic phosphate comes from the phosphate translocase. Now we have our substrates. We catalyze our reaction, we make our product of ATP, and our ATP goes back to, this is another Atp Adp translocase, right? Those are the same thing. Cool? Okay. Now, let's talk real quick about the shuttles. All the reactions that we look at in these shuttles are completely reversible. It just depends on the needs of the cell. But the whole point of these shuttles, the whole point, the reason that we have these two shuttles, so that we can maintain our supply of NAD+. Why do we need NAD+? NAD+ is what? A substrate in it is also a substrate in citrate. But why do we need to pump? The whole point is to pump NAD+ to the cytosol, right? Because we made NADH because it's used in glycolysis. So NAD+ is a substrate in glycolysis, which is in the cytosol, right? But in order to regenerate it, right? Because we went through glycolysis, we made NAD+ into NADH, but in order to regenerate that NAD+, we got to get rid of the electrons from NADH, but we can't do that because we got to drop them off in the mitochondria, but we can't get into the mitochondria, right? So we have to find a way around that. So this is the workaround cells come up with, right? So in glycolysis, start here. In glycolysis, we put NAD+ into glycolysis. What do we get out? NADH, okay? So with that NADH, right, we're carrying extra electrons. We can convert oxaloacetate into malate. Malate is something that we normally can pump across the intermembrane space, the inner membrane. Just stop at intermembrane. We transport malate in. Now we can use the malate dehydrogenase enzyme and convert it back to oxaloacetate. When we do that, what happened to, what did we do? We converted NAD+ to NADH. So we went from having an NADH in the cytoplasm to having an NADH in the matrix. Isn't that cool? Right? But the thing is, we have to complete the cycle, right? So now that we have oxaloacetate, if we convert it to aspartate, we can pump our aspartate out and convert it back to oxaloacetate so that we can keep going again. This is something that you have to remember is just a cycle, right? Do you have to know all the names of the enzymes for the shuttles? No. You just have to know that the shuttles exist and they use different substrates, and because they use different substrates, you get different amounts of energy out of them. Okay, so that's, and why do we need these shuttles? You do need to know, but that's it. Okay, so the other one is the glycerol 3-phosphate shuttle, right? So this is going to deliver electrons from NADH, but instead of making mitochondrial NADH, now we're going to use, guess what? FAD, right? Okay, so, and we know FAD goes into the electron transport chain in a different place. So that's why we get a different amount of energy, right? Because we're skipping complex 1. Okay, so same thing. We have glycolysis. In order to run glycolysis, what do we need? NAD+. We use up the NAD+ and we make NADH. That's great, but it's in the wrong place. So we use the NADH to convert dihydroxyacetone phosphate into glycerol 3 phosphate. We can transport glycerol 3 phosphate into the intermembrane space. So now we can convert that FAD to FADH2. So remember in that diagram in the very beginning, I said that there's another place that Q can pick up electrons from. This is it. So here's Q picking up those electrons and turning into QH2. Where's QH2 going? Complex 3. That's a 3. It's supposed to be a 3. Okay. So then you just have to remember it is a cycle. So we have to pump, after we convert our glycerol 3 phosphate back to dihydroxyacetone phosphate, we got to pump it back into the cytosol. Right? So this is why, but look how much fewer steps we have, right? So the fewer steps you have, the quicker you can do it. So the muscle can regenerate ATP as quickly as it has glucose, right? Which is really, really important. All right, so this is your book's summary that we just did at the very beginning. Well, not the very beginning, but you know, the quantification of where all of the ATP come from, right? But I kind of like the other one better, but you know, it's the same thing. So it tells us that in the muscle, right? Because we're going through the glycerol 3 phosphate shuttle, we only make 30, whereas in the liver, because we use the malate aspartate shuttle, we get 32. Okay, but it's the same math that we did. I just think it's easier math. Okay. Golly. All right, so when you make ATP, right, you don't always want to be making ATP. ATP is great, right? But winter is coming. So do we always want to be making ATP out of the glucose that we get? No, we want to be able to store them. So we have to have control points in all of our metabolism cycles to make sure that we're not making more ATP than we need. One of the key ways that we regulate that is through the ratio of our electron carrying molecules, right? That tells us something about the energy charge of the cell. So the ratio of NADH to NAD+ will control the citrate cycle. And that makes sense, right? What are the products of the citrate cycle? NADH, right? So the product is controlling the cycle. That's feedback inhibition. We see that over and over and over and over again. You don't have to memorize this. You don't have to memorize this. But look at the places where NADH is going to regulate. What's this straight pathway at the top? From glucose to pyruvate, we're glycolysis. So our very last step is inhibited by NADH. Then when we convert pyruvate to acetyl-CoA, guess what we're inhibited by? NADH. When we go from acetyl-CoA to enter the citrate cycle, what are we inhibited by? NADH. At multiple points inside of the citrate cycle, what are we inhibited by? NADH. What is the other thing that you see as an inhibitor of this general pathway? ATP. So ATP can inhibit, and ADP is a positive regulator, right? It can speed things up. And that makes sense because both things are telling us about the energy charge of the cell. And we want those regulation points because a lot of times these regulation points are points at which these different metabolites get shunted off to different places, right? I've made lots of ATP. I don't want to make any more ATP. What am I going to do with this acetyl-CoA? I'm going to go make some fatty acids, right? And that makes sense. We have to have stopping points in these pathways so that your body regulates what it needs and it doesn't make excess, because that's just a waste of energy, right? Okay. And then we already sort of talked about this, and I'm not going to ask you in detail. But you remember when we make ATP, there's a way to bypass the ATP synthase complex, right? By using an uncoupler. And the uncoupler allows the hydrogen ions to flow without having going through the ATP synthase complex. But there's still energy there, right? There's energy in that movement of hydrogen ions down their concentration gradient. So where does the energy go? Heat. It goes to heat. So remember when I said you see this in hibernating animals? If you look at, and it's a certain kind of adipose tissue, it's called brown adipose tissue. So if you look at them histologically, right, you see more fat droplets, smaller fat droplets, right? They're the little clear ones. Most white adipose tissue have these massively big fat lipid droplets because they're just there for storage. Here we have many more, but they're much smaller. But there's lots of red staining. This dark red are nuclei. But do you see how much more red this is than this? Why is that? You remember you like your dark meat? It's more mitochondria. So this is why it's brown adipose tissue versus white adipose tissue, because it has more mitochondria. And when we want more mitochondria, in this case, not because we want more energy, but because we want more heat. And that's what the actual activity of those brown adipose tissues will change as animals start to come out of hibernation. They'll warm up. Their mitochondria will be more active, and they'll warm up and they'll wake up. Isn't that cool? It's awesome. Okay. And I think the only other thing is, y'all know the theory of how we got multicellular organisms in general, right? That there was some organism that was engulfed by a unicellular organism. And so that's why we think in mitochondria and chloroplasts, they have their own DNA, right? That's our main driving factor for that theory. So when you look at disorders, and that's a lot of times what we do in biochem too, is we talk about it in terms of like what happens really in the body, what happens when people get sick, why, all that kind of stuff. So what you have to think about is if you have an inherited disease, right? Usually you get one copy from mom, one copy from dad, right? Well, it's not true in mitochondrial disorders because a lot of the genes that encode for mitochondrial proteins are in the mitochondrial DNA. Some of them are in the nuclear DNA, but most of them are in the mitochondrial DNA. So if you have a mitochondrial inherited disorder, it's going to have a different inheritance pattern than that of a nuclear gene, right? Because you're only going to get it from mom. Because when the sperm fertilizes the egg, the mitochondria are in the tail and the tail falls off. So you really don't get mitochondria from the sperm, you only have mitochondria from the egg. That's why. So it's very, very serious when you have these kinds of disorders because you're altering ATP production, right? And so that's kind of what I wanted y'all to know on that. And that's it. So this weekend is what? This weekend is Mother's Day. Do you remember this? Do you remember this? Did I play this for y'all? Oh, good. Okay. So it needs to load. You need to give your mom an extra thank you.
That is what I once believed. But now I know that I was wrong. You gave so much to me, Mom. Besides 1/2 a set of genes. You gave me nutrients, transcription factors, nearly everything that matters. Was my prenatal environment, reds, placental inheritance, mRNA, mitochondria, that back in the day once belonged to you. I just want to thank you for supplying them. Just like two strands of DNA are spiraling entwined, your nature and your nurture inspire the Scientists remind me that I find is true. Slightly more than half of everything I am is thanks to you. Slightly more than half of everything I am is thanks to you. Mitochondria power the cell, and they have DNA as well. Transcription factors modulate transcription. Since they're in the cytoplasm, the egg's the only one who has any sperm, I guess. They don't have the ambition. My sex determination gene means that I'm guy. On the 81X chromosome, Dad gave me one Y. Sex has over 1,000 genes, why it's less than 92. That's why slightly more than half of everything I am is thanks to you. Slightly more than half of everything I am is thanks to you. I wrote it over 9 months, I never paid the rent. Your Google reports help shape my hydrobiologic development. I took your line, sucked it dry of every nutrient. Which was true. Times I wonder where the time went. Where did it go? Sometimes I wonder where it went. I know I'll never understand all you have done for me. But since you paid for college, I'll get my BS degree. And I learned it's not BS, but absolutely true. Slightly more than half of everything I am is thanks to you. More than half of everything I am is thanks to you. You gave me nutrients, transcription factors, nearly everything that matters. mRNA, mitochondria, that back in the day once belonged to you. Nutrients, transcription factors, everything that really matters. mRNA, mitochondria, that back in the day once belonged to you. Singing the strawberry factors. We gotta get just like 2 strands of DNA are spiraling entwined. Your nature and your nurture is spiraling combined. Scientists remind me that I find that it's true. Slightly more than half of everything I am is thanks to you. Slightly more than half of everything I am is thanks to you. Slightly more than half of me is thanks to you.
I hope you enjoyed that. So don't forget to say Happy Mother's Day to your mother this weekend. That's what? That was the altar. Wasn't it awesome? It was just nerdiness to the maximum. Good luck studying.
You keep saying that, like, whenever you say, so is the glucose, like glycolysis, actually occurring?
No,
in a membrane
glycolysis
space?
occurs in the cytoplasm, but things can cross the outer membrane space, no problem.
So
They
whenever,
can't get across the inner membrane.
because like whenever you were talking about this drawing right here,
Yeah,
it just skipped straight from the cytosol to
to the matrix.
so we didn't. So it's as if we went through the inner.
Well,
membrane
now
space.
you know how these NADH are getting,
Right. I just didn't know, like, because like at first whenever you were
or no,
saying
these
this,
NADH.
I thought it meant like that's the cytosol, so like that'd be the inner membrane space
Well,
to the matrix.
You have glucose, right? You convert it to pyruvate in the cytosol. Now we can move the pyruvate
From the
from
outer
the
membrane
cytosol
through the-
to
Okay.
through the outer membrane, through the inner membrane.
So it's
to
through all the
matrix.
membranes. At first, whenever you said that, I thought it meant like glycolysis is actually.
No. No. Shut up. What's that? Okay.
I knew exactly what I missed as soon as I left. Can you explain it though? I just want to make
Okay,
sure I understand.
so this is glucose,
Yeah,
right?
yeah,
You
yeah,
named it
that's-
correctly, right? Right.
I
But
figured that out
this is
after
a
the
glucose.
fact.
This is a glucose molecule.
Yes,
Like this is why I put it on here.
yes.
So you knew that this oxygen was going down,
Mhm.
and then we're comparing it to this CH2OH. This one's going up. So an up and a down is
Trans.
trans,
alpha.
which is alpha. So this is an up and a down, which is trans
So it's
alpha.
alpha.
Then you would do the next one. This would be an up and an up, and it's beta.
I did not understand
So it's
that
just
that's what
that-
you had asked for. I thought you were talking about something completely different.
Because right, it's this oxygen right here
Mhm.
that comes in and attaches to that carbon.
Mhm.
That's why carbon's got 1, 2 oxygens, and this hydrogen gets
Mhm.
stuck here,
Yes.
right? But the question is, this hydrogen, because it's a ring,
Mhm.
is it going up or is it going down?
Yeah. That makes sense.
And it could go either way. They're both D, because
Mhm.
in the Fisher
'Cause that's
diagram,
on
it's
the
on the
right side.
right. Mhm.
But that was the part that I didn't understand, was that you were looking for the alpha and beta.
Yeah, that's the difference between the two. There are 2 ways that this could turn into a cyclic molecule.
Gotcha.
And that's why you have some sugars that have alpha linkages and some
Mhm.
sugars that have beta
Beta.
linkages. Otherwise, you couldn't have that.
Mhm. Okay. And then I was really upset because I was like, wait, did I put 2 or did I put 4?
But you only got 1 point off. But basically, I said if y'all made 2 mistakes, I only took 1 point off.
I wrote it on the back of the exam. I sat there for 10
Me-
minutes.
yeah, because I did everything. I did everything else. I did
I'm gonna double check.
everything else, and I was like, I know everything else is right except for these 2.
Oh,
I
knew I
knew
got
for a
that
fact
one
everything
wrong
was right
because I
except for
could not
these 2.
remember how to do that, and I was like, no, and I think I did good enough. That's good.
I attempted.
You did attempt.
I
You
drew
did
some-
I
attempt.
think it's a
But
good
I-
attempt.
It's a good attempt. You almost got, and I didn't take off so much on what the hydroxyls do here,
Mhm.
right? Because basically here, right? If you're on carbon 2,
Mhm.
if your hydroxyl's on this side, it goes down. So on this
Okay.
side, it goes up. But
Okay.
I didn't worry about that.
You
What I'm
were
worried about
looking
is
at it.
this and this.
That makes sense. So they're supposed to be opposite for the alpha, and then
When I
same,
worked out the first
or
time,
no,
Same.
sorry, same on the beta.
Yeah.
Okay.
was like 8
And you're supposed to do this one, not this one.
Correct. Correct,
Okay,
because in order to have been this one, what did you have to have? This is an aldehyde.
Yeah, you would have had to have a ketose.
Correct, correct,
So technically, they were right. It was just not the answer you were looking for.
correct, correct, yes.
See, this is what my brain did too, but I forgot the oxygen was in the ring. I put the oxygen
Outside the ring.
I drew these.
Then you can't have an
two,
animated
but-
carbon. You have to have an animated carbon.
I was thinking that, and then I was like, you know what? I was like, I don't feel like fixing it.
How long is the final? How many questions?
Yeah, it's like double. It's like double.
How many questions is this?
30.
40-ish? It's like 40. I can't do math. So it's 80.
But
right?
it's a different, because it depends on how many blanks and things like that. It's not necessarily,
Yeah, because the other ones weren't this long.
yeah,
They were like 20.
but there were more blanks. These had fewer blanks. So you had, it's not the number that's important.
It's the point.
Right, right, it's the point.
I think I'm gonna be okay.
Oh, you're definitely gonna be okay, close to me.
No, I can't help but be nervous about it. I just can't help but be nervous.
I only need to sign it up. But I'm still nervous about it.
It's okay, it's okay. What you got, Aaron?
I don't know. I hope I've been doing it enough, I guess. Most of it, like, I remember. Look, don't stop. I can't help it. I just hope I've been doing it enough. I can't
I'm
help it.
sure you have.
I have developed
The only thing I'm
mindset
not
that
gonna
I
even
can't
try
chill
to
that
do
much.
is the receptors.
physically cannot stress the level that you.
I don't know why I do it.
I used
I
to.
wish I would stop, but I can't make it stop. I have tried literally everything
Yeah, you're
except for
right.
medicine.
I do that.
I have tried
Good, you have
everything.
any questions?
Are you gonna be in your office later and I can look at the old ones? still?
Yeah, absolutely, absolutely. I'm going there now. I got somebody who wants to go look at an exam from another class, so you're
Cool.
welcome
I'll
to
wait
come.
for you to.
Yeah.
My only thing, did you, right here, you can talk, I don't know if it's because of this or because of these.
Because of these.
Okay, all right, because
If you, so I said one error, no, 2 errors, I took off 1 point, and then if you made more errors, maybe
Right.
it's 2,
okay,
you know.
gotcha.
Yeah.
Okay, I didn't know if it was because of that, because that's just used as a abbreviation.
Oh, I see you did. Oh.
ACO.
I was thinking, I was like, what is that? But okay, I see
Yeah,
that. But yeah, you had,
No, right, yeah, I was just curious about that.
Yeah.
Cool.
you got that stuff?
Yeah, I realized as soon as I left here, what I did wrong.
Is it
But
the
that
same
was-
one? I feel like everyone has
But
to miss
see,
that
I
one.
didn't take off the D and the L. I didn't take off on D and L, as long as you basically said it was D, because you know the D's here, but the beta and gamma are all about the hydroxyl and then carbon 6. And are they both up on the ring or
You mean beta and alpha, right?
what did I
I
say?
said gamma and beta.
Beta and alpha, yeah. I'm sorry.
It's
I'm
okay.
sorry. I'm on the ATP synthase
It's
complex.
okay, it's okay.
Yeah.
Did anybody get that question right or no?
Yes.
Someone got it right? Good for that person.
I just didn't put the D.
I just
I didn't think it
completely
was
misunderstood it.
But it's super important for your body. Your body, if you could make an L-glucose, do that for me. And then like give me a shout out on your Nobel Prize.
Oh, that's true, you did say that it's not metabolized.
Right, because we can't metabolize. It would taste the same. It would fit in the receptor, but we wouldn't be able to metabolize it. So it would taste exactly the same.
Sounds expensive.
It is expensive. We can do it. We can do it. But it's extremely expensive. So if you can make a process where we could do it at a high quantity for a cheap, oh my God, you would be a millionaire. And all I want is a shout out. That's it. Just a little shout out. That's it.
Just a shout out, like maybe
I'll see what I can do.
Okay.
How do you make your curve? So like this test.
I
got an
base it on the
curve,
average, the class average.
Bring the average to 7.
I bring up the average.
Okay.
So
that makes sense.
you can have people who score higher than 100%.
Yeah.
I got
Yeah, we
one
know.
up there going.
Dude.
Oh, you know what? We realized there was an error in your Excel spreadsheet.
Yeah, I
Yeah,
fixed
yeah.
it.
I get 35.
We had 35.
Oh, there's an error?
Well, I fixed it. But
Or I
fixed it.
downloaded it though.
So
was
download
it bringing
it
you
didn't
down or
include-
Did it bring you down or
but
up?
you could,
The error brought you up because
Because
it
it
wasn't
didn't
calculating
include,
the.
but it depends.
Oh, yeah, yeah. you're right,
Depends.
it
It
does.
depends on what you
Yeah.
make. It just didn't include one of the exams.
Yeah.
So check the
Oh.
formula that calculates the average of your exam.
Exams.
It just didn't keep.
Oh, well then
the
we
top
should,
one.
okay.
Yeah,
So it
just
depends on what your exam grade was.
Yeah.
Depends.
Yeah, I didn't do that good on the first test, so it brought me way up. Like my test average was like a 94 or something like that. Not counting my first one, when in actuality it was like an 87 with the first one.
So then mine would
So-
bring me up then because I think that was one of my.
Right, so
Like, that was
download
the
it,
second
so
highest.
download it again, or if you know Excel, just include
Yeah,
the cell
just change
above.
that.
I mean, that's,
Change that
it's
one.
not, yeah, it's not, yeah. But
I'm
y'all,
still gonna shoot for a 50. It's the
you're gonna
goal.
shoot for a 50?
Yeah.
I think
I'm
you,
probably
I
gonna do better than
think, I
50.
think you can do it. Oh, I'm still
Please.
on. Somebody asked me something on chat. I have a question about final. Ivy, are you still there?
I feel that.
Yay, Ivy's still there. What question do you have? Yes, yay, I'm sorry, Ivy.
The only thing I think I'm just not gonna even study is chapter 8, just because it's gonna take too much energy. It's just too much.
Let's see, you took your microphone off. I don't hear you.
The first couple of them.
Yeah, it's not that bad.
It doesn't look like your microphone's working.
And then like looking back at all of it is a lot easier.
Check. Okay.
Did you still have that sticky note with all the stuff that Ethan told you about the MCAT or no?
Yeah, I saw the sticky note.
Okay, 'cause I couldn't remember everything. you
Yeah,
told me,
I still have the
but-
sticky note. It's in my office. I will type it. Okay. Okay, Ivy, go ahead and type it. I'm gonna wait, so go ahead.
I know.
Y'all were awesome.
I don't know how we did it.
Awesome.
We have, for every exam, we just cram it in.
The last
For
exam.
every-
Not listening, I'm not listening. But you
Yeah.
paid attention in class too.
We haven't started studying.
See, that's what does. it
Yeah,
for
we'll
me. If
get
I
a good,
study,
we'll get a good
I
session.
will get the same.
I started
grade
studying
if I
on
study or if I
Friday
don't
because that's the kind of person I am.
Good
Right.
job.
I'm a good student.
I
not-
can never.
La la la.
What I say here in class and night is what my brain absorbs. I can study for days and it's the same thing. Like exactly the same as if I sit here and pay attention. So
That's okay. That's okay.
It was fun though.
Yeah.
I was going to
We'll
class
stop
so
the
much.
recording. Oh, thank
Yeah,
y'all.
you're a great professor. I
Yeah.
just do not